Australia contains over 6% of the world's mangroves, with nearly a fifth of its coastline covered by mangrove forests. This habitat has been subject to extensive degradation, with recent droughts destroying over 40 million mangrove trees. Much is known about the abundance of plants and animals within these coastal forests, yet one group remains undervalued in recent efforts to protect this habitat. Bryophytes are a miniature plant group made up of mosses, liverworts, and hornworts. They don't have any roots or vessels, so they can't grow out of the ground like other plants. Instead, they have to exist on other things, like rocks, soil, or trees. Due to their small nature, they have been forgotten as an important part of the ecosystem, and little is known about why they choose to live where they do. Our group aimed to close this knowledge gap. For our research project, we looked at 3 species of mangrove trees, Bruguiera, Heritiera, and Xylocarpus, and compared the bryophytes that grew on their trunks. The bark of these 3 trees are vastly different, ranging from rough and bumpy to smooth and paper-like. We expected these differences to influence the bryophyte distribution between trees.
We wanted to learn 2 things from our research. First, if any of the mangrove species has a higher number of bryophytes on its bark than the others, and second, if we would find the same or different bryophytes across the mangrove groups. To collect our data, we studied a mangrove forest in the Daintree National Park. Within this forest, we sampled 36 trees, collecting bryophytes from multiple locations on the trunk of each tree. We brought these collections back to our lab so we could use microscopes to identify them. Mosses tend to be spiky and stick out from the bark, while liverworts, on the other hand, tend to have round leaves that lay flat against the bark. After distinguishing the mosses from the liverworts, we used identification keys and the help of experts to determine their species. We used this data to conduct statistical tests to determine if there were patterns of significance within our samples.
We found many differences in the bryophyte community of the mangroves. This graph shows the number of bryophytes found compared to the number of trees we sampled. The red line with circles represents the Bruguiera trees, and as you can see, this tree species had the most bryophyte diversity. There were many unique species found in these trees, including 11 moss species and 21 liverwort species. Heritiera is represented by the green line, and this tree group had the second most bryophyte diversity, with 7 mosses and 16 liverworts. Xylocarpus is represented by the blue line, and this tree group had the least amount of bryophytes, with only 1 moss species and 16 liverwort species.
Bryophytes have been a very neglected part of biodiversity when considering the mangrove forest. It is important that they are studied because these tiny plants still play an important role in the ecosystem and add a lot of diversity to the mangrove forest. For these reasons, they need to be considered as part of restoration efforts of forests, especially for mangroves. Within our research, we found 42 unique bryophyte species, which is more than has ever been found in the mangroves before. Our study has shown that the mangroves around us have much more biodiversity than we thought before. This information will play an important role in conservation of the future. With a better understanding of the species around us, we can effectively protect this habitat going forward. We hope you will pay more attention the next time you're walking through the forest and keep a close eye on the tree trunks so you don't miss these tiny plants.